Hello everyone, this is Salipeli Situhile. My research is on the evaluation of antimicrobial resistance in bacteria associated with diarrhea in small ruminants, Botswana case study. So bacteria is commonly known as scur and is when animals have watery feces. According to Abdul Irwan 2021, it is estimated to cause about 46% of lamb mortality in the US. So showing you how very common this is. However, it is a multifactorial issue. It can be caused by hygiene issues, can be caused by parasite, can be caused by change of feed, and also can be caused by bacterial infections. And if it is caused by a bacterial infection, antibiotics are indicated for treatment. So antimicrobial resistance is when now the bacterial agent is not responding to antibiotics or antibiotics are being ineffective, basically. So what's the hypothesis of this study? The hypothesis is that there will be some resistant genes in the populations. And the main objective of this study is to identify resistance genes to sulfonamide, tetracyclines, and trimethoprim, since these are the easily accessible drugs in Botswana. Also to investigate the common infectious bacteria implicated in diarrhea in small ruminants in Botswana. So the methodology, this will be a descriptive study. What we'll do is we'll collect samples from farms, and then what we'll do is we'll carry out different bacterial tests to characterize the causative agent, and then use the disk diffusion to check for resistance genes. If we look closely at figure 1, there are two agar plates. The first plate has labels A, B, and C. So those will present the antibiotic discs. In this case, the 3 antibiotics we're testing resistance for. And then what we'll do is we will add the causative agent to the agar plate and wait for about 24 hours or even more to check for growth after it. If you look at plate number 2, around C, there's no growth, meaning that this is a zone of inhibition, meaning that the bacteria is actually susceptible to antibiotics. But if you look at A, there is growth, showing you that there is resistance. So this will give us the prevalence of the resistance in the country. So how do we get antibiotics from animals to humans? So if, say, in a case where animals have diarrhea and then we're using antibiotics irresponsibly, so what happens is that the resistant genes multiply in the gut, and then if the human eats the meat, that's how they're exposed. And also the use of dung as fertilizer exposes crops, and so the consumption of the meat and the crops will lead to AMR. So this is a very important topic. According to Hauser et al. 2021, WAR classifies antibiotics as very essential antimicrobials, which needed to be guarded and used responsibly. Also, this information can be used by policymakers, researchers, field veterinarians, and help in the possible measures for managing resistance on farms. And therefore, that's why the research is important. With that, ladies and gentlemen, thank you.